Last but not least, I'm pleased to welcome James Evans. James came to us from UC Davis in 2011. He is a specialist in interfacial. He's a specialist in protein structure and dynamics, a real hot field in the interface of biology and nanotechnology. James is currently leading our BER imaging project. James.
Thank you, Liuan. And also, I want to follow up with what Kirsten said earlier. It is a humbling experience to be invited by Don and Liuan to present at the 20th anniversary of EMSL celebration. And so today, I'm going to try and give you a high-level overview of the past for bioimaging, as well as kind of where we hope to be going. So, as kind of hopefully has been apparent through the meeting and also the tours and any interactions that you've had with EMSL, one of the biggest strengths of EMSL is the fact that we have so many diverse capabilities, both in terms of instrumentation as well as expertise, housed all in one building. And Kirsten just gave a great talk showing some of the applications from the CISA as well as the mass spectrometry and other capabilities for systems biology applications. And so the rest of this talk is really going to focus on kind of one of the other disparate areas that you haven't really heard about, primarily the electron microscopy areas and what we can do with that. But before going into really the science of that, it is important, just like Kirsten was talking about, to give some context in terms of, if you're not familiar with biological samples, what do we mean by different types of systems? And this happens to be a great example, because I'm sure many of you who came to visit wound up doing this. Every time I go on travel, I wind up going to Google Maps and looking it up, trying to identify where, say, you're staying in a hotel versus where you're going to visit. And this is an image of EMSL. So we're actually in a building just slightly off of this. But the impact here is that it's become so accustomed to us that we go between looking at satellite views versus overlays of traffic and other maps that we kind of forget the fact that this is actually a huge amount of data spanning these different scales that Google had to come up with. In fact, this only represents 5 orders of magnitude. But if you think about biological systems and what Kirsten was just talking about, biosystems actually span 10 orders of magnitude just in the spatial sense. And in addition to that, it has temporal dynamics, it has functional activities, chemical differences, and so forth. All of these things need to be overlaid on top of all these different scales for spatial information in order to really understand your system of interest. And so just like Kirsten was talking about, if you're looking at photosynthetic organisms, you have plants and unicellular green algae that are the same 5 orders of magnitude in difference that Google Maps, the publicly available information that you have from that, exist in. And so both of these do photosynthesis, both of them exist as very different biosystems, and trying to understand commonalities and differences between these to derive better ways to get biofuels or other products is really important. But this completely ignores the fact that this is 5 orders of magnitude between the whole cell size and the whole plant. If you then consider the complexity just within the cell, you have all these molecules, all these proteins, complexes, organelles that come together in order to create a rationally confined space within the cell for all these different reactions to occur inside the cell. And there's a lot of questions about how this organization exists, what are the structures of the fundamental building blocks, how do they interact with each other, how does that change over time? And this is the current state of the art, where we're starting. At the end of this presentation, hopefully you'll walk away with, we're kind of on the precipice of being able to start to answer a lot of these questions. But before getting there, it's the 20th anniversary celebration. So what was the state of the art for this type of bioimaging 20 years ago? Well, if you look in the literature and look in Science magazine, the single article published in 1997 having to do with any cryogenic electron microscopy capabilities for structural work was this article by Joachim Frank's group. And this is the ribosome. It's a multi-subunit complex that's very complicated that comes together to convert the code of DNA into proteins and enzymes that will actually be functional inside the cell in order to give rise to life. And at this time, instead of atomic resolution at 1 or 2 angstrom resolution, This map was published in Science with 26 angstrom resolution. And you can see the red donut over there with a hole in it. That hole is actually smaller than the resolution that they were able to achieve. It's 16 angstroms. And they had to formulate a hypothesis that maybe through that hole, the RNA strand that encoded the information was going through the middle of the complex and exiting through. We really didn't have the resolution to really refine that and say for sure that's what was going on. And so back in the 1990s, there was another very similar aspect with the Hubble Space Telescope going up. And the mirrors originally were not corrected for spherical aberration, and the first images came back very blurry. When NASA realized this and corrected for spherical aberration, now they wound up getting the very crisp images that we're so used to today. In 2000, aberration correction for electron microscopy became available. And this really started to herald the difference in the types of resolution that you could get for individual particles. And although material science applications really fostered this development over the first decade of the new century, biological applications have become more and more common for incorporating it. In addition to the improvement in the technology for actually how do you interact with the sample, we've had a revolution in the last 3 years where we've actually been able to utilize brand new detectors. So all the way in the last 70 years since electron microscopy was developed, you wound up working with film or with CCD cameras where you would wind up imaging your sample by having the electron hit a phosphorescent screen, create light. That light gets tunneled to a CCD camera, and you're actually recording the light. Instead, with direct electron detectors, now you can actually image the impact of each individual electron. So you have much clearer data. And because of this now, you can wind up getting truly atomic structures from individual particles and individual complexes. This happens to be from the exact same group of that 1997 article that I just showed you on the ribosome at 26 angstroms. This is Joachim Frank's group looking at another ribosome, and here you can actually resolve each individual strand of all of the individual components that come together to form this very complex structure for the ribosome. In addition, now because you have this resolution, you're starting to be able to actually ask questions about how does this multi-subunit complex come together? How is it assembled? How does that affect the overall function of it? So we're in a really interesting time for structural biology and bioimaging in the fact that now we don't have to rely on crystallography in order to take a protein, grow it into a large 3D crystal that we can get structure from. We can just take that protein and immediately start working with it to get structure. So that brings us to what do we actually have here in EMSL that users can benefit from? Well, we have a number of techniques, and starting kind of from the scale that Kirsten was just talking about, we have helium-ion microscopes up top that can take your image and of your native isolate and start to look at bacteria communities as well as yeast structures. And here, if this was an SEM image, you would have out-of-focus blur above and below. But because this utilizes helium ions, you wind up being able to image a very large structure, and everything looks all in focus and kind of has a 3D appearance to it, even though it's 2D. Very useful. This example down at the bottom is actually a slice and view image of showing the three-dimensional volume of a natural isolate. What you're seeing in orange are actually isolated cells of algae and cyanobacteria that were taken from soil, and the blue are the locations of viral particles that are infecting these organisms. And try and understand the infection mechanisms as well as the life cycle afterwards is one of the questions that the user that this data comes from are trying to answer. Taking in the next level of view, going into what's happening inside the cell, we can do electron tomography to actually be able to visualize the entire 3D structure within the cell. And we can also compare individual different mutants of different organisms in order to understand the effect of knocking out specific enzymes or genes within the organism in order to try and identify how can we design them to give rise to better biofuels. And in addition, just like the talk I was showing about the direct electron detectors, We have all these different capabilities now that with a couple of them that have direct electron detectors, that we can start to look at structures from proteins of any type of geometry that we can commonly give rise to. And so we have these capabilities, but one of the things that we've also been working on to develop is a way to make it easier for users to take advantage of these. And so instead of requiring users to become experts in biochemistry, if they're working with whole cells and they identify through some of our mass spectrometry or other omics capabilities certain proteins of interest, rather than having them have to go through trying to purify and express and purify that protein, get it to a state that they could send it to us to do structural work or functional work, we've actually created a pipeline that all they have to do is tell us the gene of interest. We will put that into a plasmid, do transcription and translation reactions in a test tube to create the protein of interest, do purification, send it to functional assays to show it's fully active, and then we can send it into our structural biology pipeline with cryo-EM here at the lab in EMSL in order to give rise to a 3D map as well as an atomic model for that structure. So this is really something that we're hoping to roll out in this next year to users. And you can see that we've actually been able to express proteins from bacteria, algae, yeast, plants across the board for BER relevance. But all of these, everything that I've showed you so far are really static snapshots. These are samples where we've either done chemical fixation or vacuum drying or frozen the sample. And even by freezing the sample, even though we can do that in a completely native environment, the act of freezing completely immobilizes it, meaning you can't get to dynamics. And so although we've spent the last 8 years trying to develop in situ holders to allow putting a sample into the high vacuum of an electron microscope and allowing it to still be in a liquid or gaseous state, so its native environment, we've made a lot of progress and have had a lot of success in the material science area. This is a movie that would be playing, but unfortunately we tried 5 different movies, 5 different codecs, none of them worked on this computer. But it would show growth. You would see individual nanoparticles, kind of like that, actually appearing from an absolutely random background. So this is a precursor solution that would grow into individual nanoparticles over time. And by being able to control the environment and as well as have it in a liquid state, in a dynamic state, you can actually track the growth of individual particles and see how they differ over time. So while that's been incredibly useful for advancing material science applications, we can put biological samples into these in situ holders and look at them. You can see a cell, a whole cell here, as well as individual isolated particles of ferritin, a protein complex. The issue is that biological samples tend to not want to be on surfaces, so they stay in solution. If you're imaging with a conventional microscope as something in solution, you have motion artifacts, tumbling, Brownian motion, other things that come into play. And you can see here that this one happened to look good. These ones were moving during the time of acquisition. And so we have this limitation for not having conventional microscopes that can operate fast enough in order to be able to look at dynamics for biological organisms yet. And so because of that, we've been focused on developing a next generation electron microscope. This one takes a completely optimized electron microscope column and integrates 2 pulsed lasers. The first one can come in, enter just above the sample plane, shine down to trigger a reaction, start a reaction. And we've designed these in situ holders to be able to be compatible with this so we can have our sample in a liquid or gaseous state. We can then vary whenever we want to for each individual experiment when we send in another laser at the very top. And we wind up sending this into a modified gun where we actually change the physics of electron microscopy. So instead of having 1 electron in the column at any given time, going down, interacting with your sample, getting collected on a screen down below on your CCD camera, and then leaving your shutter open for long periods of time to integrate and build up enough signal for many, many electrons coming down, now we actually generate a pulse of electrons that in the same time span of the laser pulse photo emits all the billion electrons that you need to form an image on the nanosecond to microsecond time scale, propagates down the system and is imaged down below. And so we actually have pulsed electrons coming out of the gun. It's fully integrated with the microscope. We have first imaging in a CW mode where it's a continuous wave as well as first imaging with pulsed waves. So this instrument is now working as a pulsed instrument, but we're trying to improve the spatial resolution and contrast and brightness so that we can start to observe some of these biological dynamics at much higher resolution. Now, this is something that is going to be coming online to the user program hopefully in the next year or so. We're trying to improve the spatial resolution, and then we'll begin starting commissioning experiments. But in the meantime, looking forward, where can we go from here? I've just talked about how we can look at structures now of individual particles. We can start to look at them inside of a cellular context. We're starting to develop ways to be able to look at things beyond a static state and get into dynamics. But the next question is, how do we actually address the original problem that I mentioned? How do we bring everything together in terms of trying to understand these critical questions of inside of a cell you have compartmentalization and different enzymes go into different compartments, and the molecules that they work on or that they produce by their reaction may be also compartmentalized in different spaces. In trying to understand both the quantity of functional enzymes, where they are in the cell, where they interact with other molecules, as well as their relation to these molecules that they work on, is really critical to this understanding. And this is actually something that I would really love to see EMSL move into in the next few years, because it is actually something that we could really build a name for. Largely because in order to make this a reality, to be able to address all of these questions, we have mass spec, NMR, and cryo-EM capabilities in order to be looking at the structures of individual complexes. So imagine being able to get a structure of every protein in the particular pathway that you care about. Once you have those structures for it, you can use electron tomography, like I showed you earlier, that we can get, where we can generate a 3D volume of an entire intact cell, looking at and identifying every single protein in its native state. We then can leverage our high-performance computing capabilities in order to do template matching and identify within this 3D volume where each one of these proteins actually are. And then generate a 3D model of the entire cell and the environment, and be able to start asking questions about what about proteins that we don't know interact but happen to always be found right next to each other? Not necessarily directly interacting, but always within a few nanometers of each other, something that no other method can currently provide. In addition to being able to answer the quantification as well as localization in relation to metabolite pools. So this is something that we actually have available to us in certain parts. Bringing it together would really harness all of the strengths that we have from the multiple different capabilities that we have across the lab, and would really push us into addressing this question in the future. So with that, I'd like to thank you for your attention, and I'm happy to answer any questions.